students, how are you all? Welcome back to my EDUspace. Hope you all are doing and learning well. If you like my videos, then don't forget to subscribe my channel and you can share to your friends as well. So let's talk about today's video. It is in the continuation of the cells of immune system. So today's video is all about the natural killer cells or NK cells. They are named as natural killer because they don't require activation to kill cells that fail to express MHC class 1 self marker. So we will do lots more regarding NK cells. Without further ado, let's get started. Now, introduction to NK cells. These are large and granular lymphocytes. As you can see in this picture, this is the diagram of natural killer cells. So these natural killer cells are much bigger in size and larger as well as granular in appearance as compared to B cells and T cells. So natural killer cells are lymphocytes belongs to same lineage as T and B cells and differentiated from lymphoid progenitor cells. So we know that hematopoietic stem cells is differentiated into two types of progenitor. The first one is myeloid and the next one is lymphoid progenitor cells. From the lymphoid progenitor cells, three types of cell lineages are formed. The first one is B cells or B lymphocytes. The second one is T cells or T lymphocytes, and the third one is NK cells or natural killer cells. The B cells and T cells are involved in adaptive immunity or acquired immunity, whereas NK cells are involved in innate immunity. They are very specific cells. The B cells and T cells are very specific towards their antigens. But in the case of NK cells, as they are innate immunity, so their main motto is to kill the cells which are abnormal or infected. Okay, so they are not as much specific as like B cells or T cells. So these cells involved in innate immune system and classified as group 1 innate lymphocytes, that is ILCs. So these are known as ILCs. Okay, as they are involved in innate immunity. They give quick response against pathogen, which is the feature of innate immunity. They give a quick response whenever they met a foreign pathogen. Okay. NK cells secrete cytokines, which are quite similar to the cytokines secreted by NK cells. T cells. Okay. These NK cells produces cytokines. Okay. They are production house of cytokines, and these cytokines are quite similar in nature, which is secreted by the T cells. They also involved in cytolysis. So both T cells and NK cells, as they are formed same types of cytokines, so they both are involved in the process of cytolysis or cytotoxic process. So they give strong cytolytic responses against physiologically stressed cells. Physiologically stressed cells means which are not normal cells. Okay. These cells may be tumor cells or viral infected cells. Now again, look at the structure of a typical natural killer cell. So this is a typical natural killer cell. They do not express CD8, TCR and CD3. The natural killer cells do not express these type of surface markers, which are CD8, TCR, T cell receptor and CD3, which are present in T cell. So they are differentiated from the T cells as they do not possess these three types of surface markers. Okay, so there is no CD8 T cell receptor and CD3. But what kind of surface markers they possess? They are having the CD16 and CD56, which help in antibody dependent cell mediated toxicity. So these two markers are help in antibody dependent cytotoxicity. Okay. They also possess two types of receptor as well. The first one is activation receptor and the second one is inhibitory receptors, which help in recognizing the self cell or normal cell and viral infected cell or abnormal cell. So these two are very important for the functions performed by NK cells. They basically help in distinguishing between the normal cell and the infected cell. So we can say that both receptors have a very significant role in the cytolysis behavior of natural killer cells. More than 2 billion circulating in our body. So the number of the lymphocytes are more than 2 billions, but they are very short-lived, large and granular lymphocytes. So these are the granules. You can see these dots are granules. So as I told you, these are large granulocytes, therefore they possess granules. That's why this is granulocytes. These granules contain biologically potent molecules that help in cytolysis. So these molecules, after activation, they get secretes out from the natural killer cells and they perform various functions regarding cytolysis. They contains enzymes like proteins or enzymes which are known as cytotoxins. Basically, these enzymes or proteins are perforins. Okay, the first one is perforins and granzyme. So perforins, as its names indicates, these proteins perforates or poke a hole. They make hole onto the cell membrane of a cell. Hence, they create hole on the affected cell membrane. Thereby, cytolysis takes place. So, what they perforin do? They create hole on the surface of membrane, which causes the bursting or releasing of all the content of the cell infected cell, and this cell is become what we can say, this cell is killed now. Okay, now what is the function of granzyme? It is basically induces apoptosis. What it does, it basically induces the apoptosis to the infected cell. Okay, so they both play a significant role in the lysis of infected cell. So we can say their granules contain preformed biologically potent molecules which help in cytolysis, and these potent molecules are perforins and granzymes. They secrete T cell like cytokines, as I just told you, but no T cell receptor is present on their surface. So how this NK cell is differentiated from the T cell, as the T cell possesses CD8 T cell receptor and CD3, whereas the NK cell possesses CD16 and CD56 molecule, and the receptor that NK cell contain is activating receptors and inhibitory receptors. which play a significant role during the distinguishing between a normal or infected cell. So now let's talk about how these natural killer cell works. Why these are known as natural killers? Because they don't want any kind of activation from other immune molecules. They just kill the cell which seems abnormal. Okay, so it just try to kill the cell which seems abnormal to it. Now, next question must be spinning in your brain, like how NK recognize normal and abnormal cell. The abnormal cell may be stressed cell, okay, which may be infected or maybe tumor cell. In other words, we can say that they provide immunological surveillance. The NK cell provide immunological surveillance. They are incomplete or all time on the watch of the cell, whether it is infected or normal. If it found infected, then it suddenly kill it by its very smart process. Okay, so these undergo immunological surveillance. So these NK cells provide immunological surveillance to our body, as these NK cells roam inside the body here and there, and suddenly as they got the abnormal cell or infected cell, they kill it. Okay, so first let me take an example of a normal cell, how we distinguish a normal cell. So this is our normal cell, okay, which having an activation ligand, this one, and MHC1 molecule, or we can say MHC class 1 molecules, as we know that all nucleated cell contains MHC1 molecule. And we also know that NK cell is having two types of receptor. The first one is activation receptor and the second one is inhibitory receptor. The activation receptor, this one, once bind with the activation ligand, okay, it's get activated or NK cell get activated. So as soon as the activation receptor binds to the activation ligand of a cell, then it becomes activated. So here it becomes activated. This become activated. Now, but here is a surprise. What surprise is that? So let's discuss. There is a inhibitory receptor here. Basically, this is very important. This one inhibitory receptor is very important for recognizing the cell whether it is normal or abnormal, as this inhibitory receptor recognize self MHC1 molecule. This is MHC1 molecule. Basically, what is the function of MHC1 molecule? Basically, they present the antigen on its surface to other cells. So here this time the MHC1, it displays normal self protein. So this time what it displays, as this is normal cell, so we can say proteins or antigen that it displays is self proteins or self antigen. Okay, then inhibitory receptor, this one, recognize this as self antigen. So it recognizes this as a self antigen. The inhibitory receptor recognize the MHC1 molecule contain the self antigen or self protein molecules. Thereby inhibition outweighs the activation. So as soon as it detects that MHC1 molecule express the self antigen or self protein, so what happened? The inhibition process outweighs the activation and NK cell recognize that, so there is no activation process takes place. Okay. So NK cell recognize that cell which I just in contact with is a normal cell or unaffected cell, and suddenly there is no activation process takes place and it just leave this normal cell. It means it could not kill this normal cell. So there is no destruction of cells takes place. No destruction takes place. Okay. I hope you understand. Let me again make you understand that we know that there is a two receptor. One is activation receptor and another is inhibitory receptor. Basically, inhibitory receptor performs or recognize whether the cell is normal or abnormal or infected with virus. Okay, so this inhibitory receptor, as soon as it make a contact with MHC1 molecule, it recognize what type of antigen or protein molecules the MHC1 displays. As this, the MHC1 molecule displays self antigen or self proteins, then the inhibitory response is outweighs the activation. Okay, and consequently no activation of NK cells takes place, which means there is no destruction of the normal cell takes place. So the idea behind killing the abnormal cell as infected cell, because abnormal cell either fail to express self MHC1 molecule or MHC1 molecule could not express on its surface during infection. So now let's see what will happen if the NK cell comes across an infected or abnormal cell, that may be virus infected cell. So here this is virus infected cell. Okay, in this case if NK cell fails to detect, here the inhibitory receptor, it fails to detect any kind of MHC molecule, there is no MHC molecule, or it detects some foreign antigen on MHC1 molecule. In case of bacteria, or there is no MHC displays on the surface, but in case if MHC class 1 molecule is displayed on the surface, then the what kind of antigen it bears or it presented, it all depends upon the inhibitory receptors. If it found that the antigen that is present on the MHC class 1 molecule is of foreign type, then the inhibitory receptor recognized that particular as foreign antigen and the inhibitory response stops and the activation process starts. Okay. So it could not inhibit the activation process as the inhibitory receptor found the MHC molecule displays foreign antigen. Okay. So here the activation receptor binds to the activation ligand of the virus infected cell. So here activation outweighs the inhibition. So what happened? There is no inhibition takes place in this case, because if there may be two cases. The first one is the cell is not expressing any kind of MHC molecule. Okay, if there is no MHC molecule present on the surface of a cell, then inhibition stops, and in case if MHC molecule is present but it displays foreign antigen, then the inhibitory receptors find it as a foreign antigen and there is no inhibition process takes place. So here the activation receptors starts activating the NK cell. Here the activation starts, which kill the infected cell via either secreting cytokines. Okay, after the activation of NK cells, there is a production of cytokines. And also these cytokines may be interferon gamma. Okay. And also by secreting these granules. As I just told you, these are potent molecules. So there is a secretion of these granules that contain perforins and granzyme that help in the cytolysis of the viral infected cell, which ultimately kill the infected cell. Okay. So we can say if a virus or pathogen is inside the cell, so this is the killing process of intracellular pathogen. So here this is also known as intracellular killing. Intracellular killing of pathogens, because here the virus is present inside the cell. You can see these viruses are present inside the cell. So this is the process of intracellular killing, where pathogen is present inside the cell. So what about the extracellular pathogen? So let's talk about next what is the process of extracellular pathogen killing. Defense against extracellular pathogen. If the pathogen is present outside the cell like this, these are not able to infect or get inside the cell, but this time they are present outside the cell. So if the pathogen is present outside the cell, in this case the cytokines, all the process is similar, the cell. So we just looked at the NK cell after activation secretes out the cytokines. Okay, there is a production of cytokines. This is basically interferon gamma. Okay, so as NK cell produces cytokines interferon gamma, which activate the macrophage. Okay, this particular process activate the macrophage, and we know that macrophages are big phagocytic cell. They are big phagocytic cell. Okay, they are big phagocytic cell. So they ingest, what they do, they basically ingest the extracellular pathogen and thereby killing the extracellular pathogen. We know the process of phagocytosis. So here is the killing of extracellular pathogen or elimination of the extracellular pathogen, or we can say the digestion of the extracellular pathogens through phagocytosis takes place via macrophages. Now the next and very important topic of natural killer cells are antibody dependent cell mediated toxicity. So we know the structure of the NK cell. It possesses two types of CD markers. The first one is CD16 and the another one is CD56. So this CD16 is the receptor of the Fc portion of immunoglobulin. It binds to the Fc portion, or that the constant region of the immunoglobulin or antibody. So this antibody is bind to the antigen. You can see this green color molecule is the antigen. Okay, so this antibody binds to the antigen, and as soon as it binds to the antigen, this antigen antibody complex, this one is the Fc portion of a immunoglobulin or antibody. As soon as it binds to the antigen, it get bind to the CD16 marker of the natural killer cell. So it binds to the CD16 marker of the natural killer cells, which ultimately leads to the destruction of these antigen which are bound on the antibody. So as soon as it binds to the CD16 marker, this NK cell causes destruction of the antigen that were bound to the antibody. This process is known as antibody dependent cell mediated toxicity. That's all for today, and if you have any query, please text me below and share your suggestions about the topics you want me to cover in upcoming videos. Thanks for watching.